Hello everyone. Today we'll talk about DNA chips and how we can use it to detect mutations in breast cancer. DNA chips are an amazing technology that can allow researchers to study thousands of genes at once in a single experiment. DNA chips can offer speed, accuracy, and specificity. They also offer flexibility because they can be designed to analyze gene expression in any organism or tissue type. All of these features make DNA chips a powerful tool for studying gene expression and understanding the molecular basis of disease. DNA chips have many clinical uses, including gene expression analysis, pharmacogenomics, infectious disease diagnosis, and cancer diagnosis or classification. Before the development of DNA microarray technology, scientists employed techniques such as northern blotting or quantitative PCR to investigate gene expression. These methods entailed the extraction of RNA from cells, conversion of RNA to complementary DNA, cDNA, through reverse transcription, cDNA amplification using PCR, and subsequent separation and detection of the resulting products via gel electrophoresis or hybridization with labeled DNA probes. While these traditional methods provide useful insights into gene expression, these processes can be complicated and time-consuming, and they have limited capacity for analyzing multiple genes simultaneously. DNA chips, on the other hand, allow researchers to analyze thousands of genes at once, making them a high-throughput approach to gene expression analysis. So, what are DNA chips? DNA chips are small devices that have many tiny probes of DNA, and each probe can identify a specific DNA sequence. It's like a small piece of glass or a silicon chip with a bunch of tiny spots arranged in a grid. How are DNA probes planted onto the chip? DNA probes can be made on DNA chips using a process called photolithography. This process involves creating a pattern of light on a photosensitive material, which is then used to create the DNA probe pattern on the chip. Here is a general overview on photolithography. First, the chip surface is coated with a photosensitive material. Then, a mask is placed over the photosensitive material and exposed to ultraviolet light. This mask is created with the desired DNA probe pattern, and it contains transparent areas where light can pass through, and opaque areas where light cannot pass through. The areas of the material that are exposed to light become chemically altered and are able to bind to DNA. The specific order of sequence synthesis on the wafer surface is determined by applying distinct lithographic masks sequentially. During each chemical synthesis cycle, UV light serves to remove the protective groups covering the array surface. This step enables the addition of a single protected nucleotide, which is applied by washing it over the microarray. Repeated cycles involve successive rounds of UV light deprotection, adjustments to the filtering patterns of the masks, and the addition of single nucleotides. This iterative process results in the creation of microarray features, each with specific 25 base pair probes that are synthesized with a specific sequence that is complementary to the target DNA. These fragments are attached to the modified areas on the chip surface. When the chip is exposed to a sample with the target DNA sequences, these sequences will bind to their corresponding probes on the chip, allowing the DNA to be detected. The size and number of spots that contain probes can vary, but most chips can hold thousands to millions of probes. There are also different kinds of DNA chips that use different ways to show the results, like using one color or two colors. In this way, researchers utilize fluorescence. To prepare a DNA chip labeled with fluorescence, a small amount of the sample containing the target DNA sequences is first extracted. Next, the DNA sample is labeled with a fluorescent dye, which can be done using a variety of methods such as nick translation, random priming, or PCR amplification with fluorescently labeled nucleotides. Once labeled, the DNA sample is then hybridized to the DNA chip, which contains many small spots, each containing a specific DNA probe sequence that is complementary to the target sequence. During hybridization, the labeled DNA sample will bind to the complementary probes on the chip. The chip is then washed to remove any unbound DNA, leaving only the DNA that has hybridized to the probes. To detect the hybridized DNA, a fluorescence scanner is used to measure the intensity of the fluorescent signal emitted by the labeled DNA on the chip. The intensity of the signal at each spot on the chip corresponds to the amount of hybridized DNA, allowing researchers to determine which probes have bound to their target sequences. So now we understand how DNA chips work. How do they apply in breast cancer detection? When it comes to breast cancer, scientists use DNA chips to find out if there are any changes in two genes called BRCA1 and BRCA2. These genes can make it more likely for someone to get breast cancer. BRCA1 and BRCA2 are genes that produce proteins that help to prevent the growth of cancer cells in the body. However, when there are changes or mutations in these genes, it can increase the risk of developing breast, ovarian, and other types of cancer. Individuals who carry mutations in the BRCA1 and BRCA2 genes have a significantly higher lifetime risk of developing breast and ovarian cancer compared to the general population. For instance, women with mutations in these genes have a lifetime risk of up to 72% for breast cancer and up to 44% for ovarian cancer. So, if one person comes up to a doctor and wants to find out whether she carries mutations in BRCA1 or BRCA2 genes, what are the steps? First, doctors collect a small piece of a patient's DNA, usually from their blood or spit. Then they make copies of that DNA using PCR. Next, they label the DNA with special tags or markers that make it easier to visualize on the DNA chip. The labeled DNA is then placed on a DNA chip that has thousands of probes that match specific parts of the BRCA1 and BRCA2 genes. The labeled DNA sticks to the probes that match it on the chip. Then, the microarray chip is scanned to detect and record any variations in the signals emitted by the labeled DNA at each probe position. In cases where the individual lacks a mutation in the gene, both the red and green labeled DNA samples will successfully hybridize with the DNA sequences on the chip that correspond to the normal or unaltered gene sequence. Conversely, if the individual carries a mutation, their DNA will not effectively bind to the DNA sequences on the chip that represent the normal gene sequence. Instead, it will selectively adhere to the chip's sequences designed to match the specific mutated DNA sequence. This distinct binding pattern indicates the presence of a genetic mutation in the individual's DNA. Finally, doctors compare the results to a database of known changes in the BRCA1 and BRCA2 genes. This helps them figure out if the person's DNA has any changes that could put them at higher risk for breast cancer. Presently, DNA microarrays find applications in clinical diagnostics for certain diseases. They are also employed to assess which drugs may be most suitable for individual patients, as our genetic makeup plays a crucial role in determining how our bodies metabolize these medications. With the emergence of advanced DNA sequencing technologies, some of the tests that previously relied on microarrays have transitioned to using DNA sequencing. Nonetheless, microarray tests remain a cost-effective option comparing to sequencing, making them viable for extensive research studies and certain clinical examinations.